thank you both. The next speaker is Bjorn Alsberg. So
Three. Hello? Yes. Mm-hmm. Okay, thanks for the introduction. Hello everyone. I'm going to start with a quotation, and it's from an article in MIT Technology Review from 2011. It says, new materials are critical components of emerging technologies that promise to be major growth areas for economy, but the journey from new material to product typically takes 1 to 2 decades. So obviously that is too slow. We need to speed things up and make more efficient progress. So how can we do that? And one source of inspiration is from something called materials informatics. That seems to become more popular in material science in general. And it's a field where machine learning and computer science methods are being used to facilitate the discovery and development of new materials. And this field is inspired by similar fields from chemistry and biology, such as chemometrics, chemoinformatics, bioinformatics, and systems biology. And my question is, how can material informatics be applied to organic electronics? So what I'm going to present to you is our take on this. And our take on this is to use artificial evolution. And the whole idea is that you use the computer to automatically assemble molecules, and you do it in a Lego-like fashion by selecting fragments from a database, and you select the fragments from existing molecules because you want to have realistic possible structures. And then these structures are iteratively refined in the computer according to the principles of evolution by optimizing a fitness value. It might be several fitness values, but the fitness value is reflecting material property of interest such as refractive index, the power conversion efficiency, and so on. So here you see the steps in the artificial evolution. You start with initial population of structures. So this is like summarizing your state of knowledge. Typically you will start with some kind of experimental existing molecules. And then you do some fitness evaluation on these structures. You obtain the fitnesses. And you have a termination check there, but if you're okay, you move to the next step, which is then to select a subset from your initial population. And the selection is based on the fitness value, so the more fit the molecules, the more likely they're going to be selected in the next step. Once you have this subset of structures, you perform evolutionary operators to generate new structures. And these evolutionary operators are crossover and mutation. I'll show them in the next slide. And this brings to a new population of structures. So the whole process can be imagined like a flock of birds, a swarm of bees or whatever, over a fitness landscape, and you're trying to localize the global optimum of the fitness. So evolutionary methods, they are global optimizers and not just local optimizers, which are the more general, more common method. So the evolutionary operators, as you know from biology, you have genes, and that contains the biological information contained in a linear fashion to give you chromosomes. But for molecules, we're not using a linear data format. We are using actually the graph structure of the molecules. So here you see typical examples of crossover and mutation operators. To the left you have two parent molecules, and you're swapping fragments on selected positions to generate offspring structures. And you also have mutation, where you go into a certain place and just random select or change a new fragment. So the most important part maybe of this process is the fitness evaluation. So how do you do that? The three main sources of fitness: the first one is you can do synthesis of every structure you find and then test it. You could do quantum chemical computation of every structure, or you could do machine learning models which are trying to predict from structure the property of interest. And each of these approaches have their advantages and disadvantages. So I made a little table here to summarize. So if you look at the experiment, it will have a high accuracy but a huge cost. I would say completely impractical, because the evolutionary run will typically go through thousands of structures. So it's not really practical. And I made two other columns there. One TU means that do we need to have a theoretical understanding of the relationship between the the structure and the property of interest? No, we don't. We can still do the experiment. And the last column is related to novelty, meaning that what if you have structures you've never seen before? You can still obviously do an experiment. When it comes to quantum chemistry, you have moderately to high accuracy, but it could be at a very high cost depending on the level of accuracy you need, because there are many different types of quantum chemistry methods. Some of them are cheap, some of them are okay, but then you sacrifice for accuracy. So if you want high accuracy, it could be extremely difficult to do. So what about theoretical understanding? Yes, you have to have that in order to make the prediction of your property. The novelty, meaning that what happens if you have completely new structures, that is tending to be good, but there are exceptions. DFT needs to optimize the functionals and things like that. And then you have the machine learning models, and they tend to have moderately to high prediction accuracy at a very low cost computationally. So that's good news. And the other good news is that you don't really need to have a theoretical deep understanding of the relationship between the structure and the property, not necessarily. The more you know, the better it is, obviously. The problem becomes the novelty, because if you have completely unseen structures, then the machine learning models will tend to have a problem. So that's what we call the extrapolation problem, how to make predictions for completely unseen novel structures. But the conclusion is that we want to use quantum chemistry whenever possible, but if not, we use machine learning. In many cases we see that it is actually necessary to use machine learning. So let's have a look at some of the steps involved that are needed to do machine learning in this case. First of all, you need a sufficient number of structures which contain the true fitness, meaning that the true property of value for the molecule determined either from experiment or from very accurate quantum chemical calculations. And a very important point here when it comes to use of experimental data is that the experimental conditions need to be comparable, because we have seen that we're going into the literature that there's a lot of variation and you need to know about it, otherwise that's going to affect the variation of the responses. And then the structures will be used in two different ways. The first one is model building. That is the calibration set. That's where the actual model is being constructed from. And then the other part will be for model validation, meaning that you want to use independent structure for determining the predictionability of the model. And then you need to find a suitable representation for the molecular structure. There's a huge area on that. And you also need to select what type of machine learning method you're going for, and it's going to be either linear or non-linear. And at the end here, you see this thing about new structures, and the model will be only valid, locally valid, and that is the applicability domain or the confidence interval, confidence region. So if you're trying to predict within the applicability domain, then that's okay. But once you are outside, you have a problem, and you need different warnings that tell you how far outside you are. And this is something called applicability domain analysis, ADAN. So if you look at extrapolation, because this is a problem we need to address, especially now we're trying to use machine learning to be used as fitness providers in an optimization setting. So we're using evolution to optimize a property, and obviously we want to move away from the fitness values we've seen so far. We want to move into the unknown, and obviously beyond what the machine learning model has seen. So that means we need to be careful. And the two things to do to handle this problem. The first one we need to know through warnings, and the other thing is to do updating. So here I made a very simple example where you see for just one dimension, and you can see here are the calibration objects. So if you are using the model within that region, then that should be okay. And you see that this is the blue true function, and this is the red machine learning approximation. So within that region of the calibration objects, things are okay, but the problem becomes when you're moving outside. So when you get structures outside, preferably if you're maximizing the property, you will be somewhere here. But the problem is, how far can you go? Because as you can see, these two functions will diverge, and hopefully in close relation to this boundary, you could still apply the machine learning model for extrapolation. But outside, again, this is going to be a fuzzy boundary, but you are the one responsible. So you're using these ADAN measures to tell you you're really, really far away. Do you really want to use these fitness values or not? All right, so that brings us to the other big problem, and that is representing the molecules, because all of these methods, the machine learning methods, I would say 99.9% of them require a vectorial representation of the object in question, in this case a molecule. So that means you need to turn this three-dimensional complex object into a long vector which is resembling what you want to work with. So this is then an n-dimensional space. So the molecule is just a point in this n-dimensional space, and each of the dimensions represent what we call structure descriptor variables. And these structure descriptors, they are capturing some part of the structure of the molecules that are hopefully useful to describe the problem. So example descriptors, atomic charges, distances, HOMO-LUMO gaps, whatever. So it's a huge variety of descriptors out there. We have worked with a certain type of descriptor which is basically satisfying two very important requirements, because if you start with this business of creating structured descriptors, you will quickly realize that you can't just use a simple coding of the 3D structure of the molecule. So you need to do something to make them alignment independent. So if you heard about a method called COMFA, it will grid the space of the molecules and use each voxel as an individual variable. That is possible, but then you have the problem of making sure the molecules are aligned correctly and comparable. And so comparability means basically that each variable has the same meaning for every structure. And that is extremely important. If you violate that, all of these methods will not work. So we have worked with something related to a density distribution like a descriptor, where you have, let's say, a series of numbers that can vary in size for each molecule, and the question is, how do you compare these series of numbers with each other? And what we're using then is this density distribution instead of the numbers directly, and then we sample this function, and each bin that we sample will then become a variable. And it turns out that this type of approach is very effective. We're able to get quite good models using this approach. So we have two examples here. Eigenvalue descriptor, which is based on the vibrational frequencies of each molecule. And so you have a series of frequencies, and then you do this kind of a histogram or distribution function, and then you can create your descriptor. And you can also do it for orbital energy levels. It's not limited to that, so it's a general approach. Alright, so here are our current application areas. The first one is dye-sensitized solar cells, and we have also applied it to CO2 capture using amines and ionic liquids. And the last application is something we just started 3 weeks ago, something like that. Yeah, so that's very preliminary. So dye-sensitized solar cells is a non-silicon based solar cell where the dye here is of central importance, and this is a dye which is attached to this mesoporous semiconductor layer and is responsible for injecting the electrons into the solar cell. So that's being excited by the photons, and the question is then, how do you optimize these solar cells? And you can do a lot of tweaking with the device itself. Obviously you can change the electrolytes here, you can change various settings, but it's very important to get a correct dye molecule. So that's what we've been doing. We've been concentrating on trying to propose better dye molecules using the method I just described to you. So in this first example, we looked at a series of phenothiazine dyes, and you see one example to the right, and we decided to use the power conversion efficiency as the fitness value. And immediately we ran into a problem, and that was how do we get that? We cannot really do quantum chemistry to predict the PC directly. You can predict certain properties that are correlated with the PC, but you can't do the full computation yet. It will probably arrive. So at the start we were completely dependent on forming a machine learning model. So we had to use literature data, and it was very important to have curated data to make the experimental conditions as comparable as possible. So we used actually 59 dyes to form the calibration set, and the range was between 2 and 8% in the PCE, and the predictability was R² 0.7. It could have been better, but that was what we had, and we asked ourselves, could we use our method to generate or propose new structures that have predicted PC values more than 8%. So here's a snapshot of the evolutionary process, and you can see you have two parent type of dyes that are mating, and they generate an offspring, and you can see that the fitness of that offspring is actually less than the parents, but when it's mating with another structure, that offspring has the highest of all of them. So that just shows that you should not use an elitist type of selection. You need to have some low fitness individuals in there as well. So this is a visualization of the structure space, and this visualization contains both the calibration set from the experimental data and the structures generated by the algorithm. So what you see here at the lower left, let's see, yeah, there we are. Here are the calibration data, and the rest is then generated by the algorithm. And the PC values predicted, it's important, is going from lower left to upper right. So obviously we would be interested in these structures up here. And this work has been done mostly with, by my researcher, Vishwesh Venkataraman, is sitting there. So he's doing all the heavy calculations and model building. And we have then selected 3 of them to just inspect, and these are 3 structures with predicted PC values, as you can see, 9 to 11, but they also have quite large uncertainties. And this is again coming back to this problem of extrapolation. The ADAN here is giving off alarm bells. The more bells, the more you have to consider noise and variability. So in this case you can see you have 2 warning bells, but up here for the 11 it's a lot of warning bells, you're really far off. So the question is, do you want to accept this or not? This is something you have to accept or not. So at this stage it's very difficult to tell is these real structures, so we need to obviously get experimental verification. And the essay here is synthetic accessibility as a score that can be made theoretically to say something about the expected number of synthetic steps, I think it is. The higher the number, the worse it is. Isn't that true-ish? Yeah. Alright, so the other application area that we've been working with is carbon capture, and we are talking about chemical capture using different types of absorbents. And in this case there are several properties that need to be optimized for having a good absorbent, and one of the most important is the high pKa value. But in addition to that you need a lot of other properties to be satisfied in order to have a good absorbent. So what we decided was to perform machine learning models of all of these properties, but concentrating on the pKa value prediction and using that as a fitness. So we did the evolutionary run using the pKa as the main fitness, but using the other QSPR, sorry, I'm using an old word, machine learning models to filter out undesirable structures. So that's another way to handle multi-objective optimization, because we have actually more than one fitness and there are different ways of doing that, and in this case we are optimizing with respect to one important fitness and then using the remaining machine learning models to take away those that are not satisfying certain ranges, because you're not supposed to optimize in terms of getting as low density as possible but the density and viscosity has certain sweet spots in terms of where they should be. So that's why the idea of using filtering is such a good idea. In addition, we also perform density functional theory on the pKa calculations to have some kind of independent validation of the predictions. So in this case we tried out on imidazoles, which are promising candidates for CO2 absorbance, and 102 structures were being used to form the calibration set, and the pKa range was between 3 and 9. So we wanted to, as similar for the previous example, to generate structures with higher than 9 pKa values. And these are the top 21 ranking structures that we generated using the evolutionary scheme. And what you see here is a comparison with DFT. And don't believe that DFT is necessarily much better than machine learning here, okay? Because the performance of DFT in this case was very disappointing. I was surprised by the poor correlation between experimental pKa and the DFT. But it was a way to give us an additional, completely independent verification or validation of what we've been doing so far. And what you see here is that, well, first of all, the good news is that we managed to push most of the structures beyond the maximum pKa seen in the calibration data, which was around 9 or something. But we also see that as the number of ADAN violations is increasing, then it also makes the difference to the DFT predictions much bigger. So you can see those in the red here. We have more than 3 ADAN violations, so 3 alarm bells are going off, more than that. And you can see that it's quite a lot of discrepancy between the DFT calculations and the machine learning predictions. Again, it's Vish doing all the computational work here. So far I've been showing you what we can do in terms of proposing new structures that may or may not be better than what we've seen so far. So it's very important to stress that we're not claiming these are really better, but these are structures that, using the information we have and the modeling we have, these may be interesting to pursue. So in order to move further, we must do experiment, and therefore we have initiated collaboration with our colleagues at the department, and Bjorn and Oddreide, so you heard his talk. They are involved with the DSCC part, and Audun, Bjørn and Solon are involved in synthesis and also testing, so they're building up a lab there. And Professor Anne Fiksdal, she's involved in the CO2 capture, and we also investigate the ionic liquids. And Sigvart Evjen, he's the PhD student doing all the synthesis on that part. So we're very excited to do this, be part of this collaboration. All right, so the last part is a little bit rough, but I just wanted to show you what we have in mind. And this is something after having a discussion with John when he was here last time, so that's why I show you this today. Yeah, so we talked about what other properties could we be modeling and work with in terms of the evolutionary optimization, and the question was, could we optimize the refractive index of polymers, because that would be interesting in many different applications. So we know that we can use quantum chemistry on the monomer unit to get a very good estimate of the refractive index. But the problem is that if you want to do high throughput screening through thousands and thousands of molecules, it's going to be very costly. So therefore we have performed a machine learning model. And the model was based on fairly small number of monomers, 180, and used something called partial least squares regression, and the results are quite good. It's 0.9 R squared for both the training and the testing data set. So in order to test this further, this is again Vish doing this work. He downloaded monomers from the Harvard Clean Energy Product Database, because we just needed some example structures to play with, and then just fed 10,000 structures through the ML model, and you can see the histogram distribution there. So it was very. How long did that take, Vish? Can you? 10 minutes. So that's quite promising. So the next step that we're going to try with this project, this is very preliminary results, is to do the evolutionary optimization to see how far could we push the refractive index. But in addition to that, we also want to add these other machine learning filters to account for optical transmittance, suitable thermal decomposition temperatures and so on, that are desirable regions for other properties, because we want them to be interesting as optical interesting polymers. That's where we're aiming at. So that was just a quick summary of something we are working with, and I would like to, before I end, say something about funding. First of all, I would like to thank the department for funding this meeting, the COREM meeting. Without our department, we would be doing nothing. And also thank the department for funding several people working with the DSSCs in the department, and that's also been very critical. And also thank the Research Council for originally funding the development of the method, because we started that actually for catalyst design, and now we also have a grant for carbon capture. And of course the NOTU is thanked for allocation of computational resources, and I thank you for your attention.
Do we have any questions for Bjorn?
So you showed this graph on where you compared DFT values to your predicted values using machine learning.
Yes.
Okay, so first, when it comes to the DFT method, what kind of molecules was this, and did you run a sort of first principles MD to sort of simulate the complex interaction?
No, it was DFT all the way. I don't think we did any molecular dynamics there, no.
No, but this would be DFT MD, so...
No, it was not DFT MD, no.
Okay. Because when you're talking about pKa, is that relative to water? So...
Yes,
okay,
yeah.
Right. Because water has a very complicated chemistry for DFT.
Mm-hmm.
And one might need to sample a large part of phase space in order to get the thermodynamics correct.
But I don't think that is being done here. We have some solvent modeling. I think it's continuous modeling, SMT8 I think it was, being used here. Yeah.
Okay, okay, right. The other question is, do you ever find that even if you have a low ADAN value, that the method fails completely, or can you sort of...
Yes, that is possible, yeah. Because the machine learning model, even if you're within the confidence region, you might actually have a poor model, and you could see that, how much was it? 0.7, something like that, yeah. And in R squared, so it wasn't brilliant. Yeah.
Thank you. Extremely interesting talk. The fact that someone's even trying to predict PCEs in the solar cell, to me, is unbelievable, because especially something that's a combination of so many different things. How does sort of the system perform if you use it for something maybe more specific with a subset? So for instance, for ionization potential plus maybe excited state lifetime, much more measurable things, which would then sort of not so reliant on 20 different things all coming together.
We have a paper where we have the voltage times the current, and that seems to work quite well. And we also confirmed in this work the electron injection efficiency, which can be computed using DFT. So that was being done as a kind of independent validation, not exactly the same as PCE, but giving us an indication that we are on the right track, yes. So those values were in the limits that we expected, yeah.
Could you maybe say something more about how you calculate the synthetic accessibility?
Yes, I'm sure that Vish can tell more about it, but I think it's related to a calculation of the different, the number of different fragments. Isn't that true? That's like a...
In fact, to kind of elaborate further, what we have done is to take zillions of molecules from PubChem, which basically contains quite a few synthesized structures, and fragment it. And there are several different fragmentation schemes that you could use. And the frequency of the fragments is then taken into consideration, and you can actually get a score out of it, which ranks it from 1 to 10, and it gives you the scale of, or ease of synthesizability. So if you just look at the frequent fragments, or the greater the frequency, the more synthesizable it becomes. That's simple, very simple synthetic accessibility score.
Okay.
Maybe I'll ask you a question.
Yeah, sure.
I have a piece of advice for Vish, actually. Yep. Never admit to your boss that what looks like a humongous amount of work took you 10 minutes.
But it was a computational time, not the actual work. The manual work was much longer.
So I have one question, which is obviously using genetic algorithms sort of translates very naturally to the kinds of things you're doing. But I guess you alluded to one potential drawback, which is you end up exploring lots and lots and lots of potential combinations. Have you looked at using other optimization routines that are geared towards optimizing expensive functions, so things like response surface methods? I don't know if that's practical here.
Yes, we're aware of that. The problem is to go from the descriptive space and the real structure space. The problem is that if you're trying to use response surface modeling, then the problem is that how do you go back from an optimum and back to a molecule? Because there's no inverse.
Okay.
Yeah, so it will also be difficult to actually move in this space, because if you have, let's say you are in the descriptive space and you say let's move in this direction, well, how do you actually do that? Because you need to actually make molecules that correspond to those positions in the structure space. We are actually currently trying to make a method to do more or less what you're asking for. We call it like it's something called inverse QSPR, quantitative structure activity property relationships. That's what we call machine learning, because QSPR is so unattractive. It's an unsexy word. It's difficult to say. So machine learning is now everything. And what we're trying to do there is to use an existing partial least squares regression model and use that to guide the search more directly, because as you correctly observed, when you're using genetic algorithms, you're exploring quite a big space, and you actually know a little bit more. You could have directed it a little bit better, because if you have a machine learning model between structure and the property, you actually know a little bit about where you should go. And why not use that information? So that's what we're trying to do with this new thing, but it's not been published. It's still in the making. But it's a very good question. But a lot of this is related to the problem that the structure space, the real molecular structure space, is very different from the structure descriptive space. And there's not an inverse back and forth between those two spaces.
Interesting.
Yeah.
Thank you. Do we have any other questions for Bjorn? If not, can we thank all of the speakers?